Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. We were discussing beneficial microorganisms in food, and in this lecture we will be discussing different sources of microorganisms. In this lecture we will be discussing the different sources of microorganisms from soil, water, air, plants, animals and sewage. So, the microbial flora of a food consists of the microorganisms associated with the raw materials, those acquired during handling and processing, and those surviving any preservation, treatment and storage. Since these microorganisms do not arise by spontaneous generation, they must contaminate the food at some stage of production, harvesting, handling, processing, storage, distribution or preparation for consumption. Most foods are subjected to many potential sources of microorganisms. Then why should we be connected with sources of contamination? Primarily so that we can control contamination and keep the microbial load on or in the food as low as possible. By doing this, we obtain a longer shelf life for the food. We hope that this reduces the chance of microbial foodborne illness when the food is ingested. By keeping the contamination low, we can more easily control or eliminate the microorganisms with food preservation techniques. The potential sources of contamination are soil, water, air, plants, feed or fertilizer. Animals, human beings, sewage, processing, equipment, ingredient, product to product and packaging materials. Microorganisms can be exchanged between these sources. For example, animals contaminate the soil with fecal materials. Then rain washes the microorganisms into the creeks and rivers. This water may be used for irrigation and contaminate plants used for food. Thus, although water is the carrier, the microorganisms originally come from animals. For some foods, it is difficult to determine how many of the organisms in the flora are contaminants and how many are the result of multiplication on or in the food. The adjacent figure shows that the potential sources of emerging pollutants like pharmaceuticals, cosmetics, pesticides, herbicides, plastics, personal care products and many others. These will contaminate the agriculture. From this agriculture, it will go to the household, and all these chemicals again it will be goes to the hospitals. From here it will go to the waste. From household and hospital waste they will again use for the landfill, and from landfill it will contaminate the surface water, then groundwater, and then ultimate it will go to the consumers. The industrial waste in the form of smoke that will fall down again with the rain, and in the form of waste is effluent, it will go to the water bodies, which will either go to the livestock. Again this livestock, when it will be consumed, it will go to the household, or when the water bodies are used for the irrigation, again it will go to the water bodies. So, this is the chain completes the pollution of the food material. The sources of contamination as per the FSMA, which stands for the Food Safety Modernization Act, which is a law passed by the US Food and Drug Administration in 2017, that actually applies for both human food as well as the animal food, including the pet foods. The main sources of contamination is the human. The human can spread pathogens through fecal material, saliva, mucus or other bodily fluids, usually with improper hand washing and hygiene practices, illness or injury. The another important source of contamination is soil. The raw manure used as a soil amendment can pose a risk to fresh produce if not properly handled or applied, such as applying too close to harvest. The another important source of contamination is animals. Both wild and domesticated animals can spread contamination through their feces. The another is water. Water can carry and spread pathogens, contaminating large amounts of produce through irrigation, crop sprays, washing and cleaning, etc., flooding, etc. The another is the equipment. The equipment, tools and other food contact surfaces that are not regularly cleaned and sterilized can harbor pathogens and pose a huge risk to fresh produce. The from soil. Let us go into details. The from soil. Soil is the natural habitat of many microorganisms which at times may found there in high numbers. The microbial density is greater nearer the soil surface than at deeper levels. The types and numbers of microorganisms vary with the type of soil, that is sandy or clay, as well as with environmental conditions. The environment is changing constantly, especially moisture and temperature. The microbial growth in soil is limited to areas of organic material. These areas include the roots of plants, plant debris falling onto the soil, animal carcasses, fecal deposition by animals and dead microorganisms. Besides the animal carcasses, on the soil surface, dead earthworms, insects and other small animals are in the soil. This is presented in this figure, like the contaminants or the degrading bacteria, they will grow on the debris of the plant materials which are fallen on the ground, and this will again eaten by the earthworms, and they will again release to the soil, and this again it will be transmitted in the form of microbial and rhizosphere degradation, then it will be taken by the plants or it will be taken by the animals, and again the insects and other sea animals, it is eaten by the chicken and ingested there, and from there it will go to the human being. The growth of microorganisms is influenced by the chemical composition of the material undergoing decomposition. The rate of decomposition of the chemical constituents of these substances and the environmental conditions, fats, waxes and lignin may gradually accumulate in the soil because of their relative resistance to the decomposition. This residual, called humus, is subject to slow and gradual attack by a variety of microorganisms. The main factor affecting multiplication is nutrient depletion. In general, the soil environment is not a good medium for the growth of microorganisms. However, they tend to survive. Now, types of microorganisms in soil. Due to the usually poor environmental conditions, microorganisms in soil are in some form of resting state or in the reduced state of metabolic activity. Spores of Bacillus and Clostridium are quite prevalent in some soils. Generally, bacteria outnumber other types of microorganisms. Those that are common in food and found in soil include Acinetobacter, Alkaligenes, Anthobacter, Bacillus, Clostridium, Corynebacterium, Flavobacterium, Micrococcus, Pseudomonas and Streptomyces. Yeasts are prevalent in the soil of orchards and vineyards. Molds, especially spores, are found in some soils. The natural habitat of Clostridium botulinum type A and type B in soil. The natural habitat of Clostridium botulinum type A and type B is soil. Clostridium botulinum type E was not found in cultivated soil or woodlands. Thermophilic spore formers that can spoil canned foods are found in soil, usually in low numbers. Enteroviruses survive sewage treatment can be found in soil on which treated wastewater is discharged. The viruses are absorbed to soil particles, which makes them relatively resistant to inactivation. Another important factor that influences inactivation is temperature. At 4 degrees Celsius, poliovirus persisted at least 180 days in saturated soil. Other microorganisms become inactivated in soil by predators, bacteriolytic enzymes or toxins. In dry organic soils, Ustilago spores remain viable for 1 year, but in wet organic soil they should remain viable for less than 1 week. St. John and Matches in 1982 reported survival of Clostridium perfringens for at least 24 weeks in tilted soil. Now, contamination of food. The microorganisms in the soil can contaminate tubers or root crops by direct contact. Also, dirt is blown by the wind or is splashed by rain falling onto the soil, so that the dirt can contaminate crops such as strawberries, beans, cabbage or peas that grow near the ground level. The microbial numbers and types on crops are influenced by the degree of contamination of the soil in which they are grown. The numbers are usually higher near the shore than the deeper levels. The bacteria found in marine sediments include Aeromonas, Bacillus, Chromobacterium, Citrobacter, Escherichia, Pseudomonas and Vibrio. These sediments serve as a source of microorganisms for water as well as for fish and shellfish. During trawling along the bottom with nets, the sediment is disturbed so that it contaminates the fish or shellfish that are caught in the nets. Now, from water. The water is a potential source of microbial contamination of food. Rain contains microorganisms are washed from the air. As the water lands on the ground, it is further contaminated by soil microorganisms. In the ocean, organisms are in greater abundance near the shore than in regions distant from land. The dumping of wastes such as sewage and the runoff from animal feedlots results in considerable microbial contamination of waste of waterways with enteric types of bacteria. The adjacent figure shows that the receiving water, that actually the wastewater treatment from it is coming from hospital sewage, it is coming from animal waste, all these human waste, all these things are coming and contaminating the water tanks, etc., from where it goes to the human being. Now, the types of microorganisms found in wastewater. The genera of bacteria that are tend to be part of the natural flora, part of the normal flora of water include Pseudomonas, Flavobacterium, Cytophaga, Acinetobacter, Moraxiella, Aeromonas, Corynebacterium, Streptococcus, Klebsiella, Alkali genes, Bacillus and Micrococcus. Also, enteroviruses are quite resistant to the treatment given to sewage and therefore can be found in water supplies. Escherichia coli is used as an indicator of fecal pollution of water in temperate climates. This organism supposedly dies rapidly in the relatively hostile environment of rivers due to low temperature, sunlight, toxic chemicals and a lack of nutrient. However, some reports indicate that the coliforms and E. coli seem to maintain their pollution. Water is still the main carrier of organisms that cause gastroenteritis in humans. The contamination of food. Water contacts food during production, harvesting and processing. If the water used for irrigation of various crops is contaminated with sewage effluent, the fruits and vegetables can be potential health hazards. Seafoods are harvested from water. The microorganisms in the water contaminate the surface, gills and intestinal tract of fish and shellfish. The occurrence of fecal coliforms in fish is a reflection of the pollution level of their water environment. When bivalve mollusks feed, they filter large quantities of water and concentrate bacteria and viruses that are present in the water environment. Shellfish are normally found in water near the shore. The water is subject to contamination of runoff water carrying soil microorganisms and sewage outfills. Potential pathogens in the drinking water of animals can be a health hazard to the animals and to people who work with the animals. These organisms from infected animals also can contaminate the carcasses during the slaughter operations. During harvesting, water may be used for hydrocooling of vegetables. Irrigation or surface water that is polluted with sewage or animal waste can serve as a source of inoculation with various organisms including potential pathogens. Since many vegetables and fruits are eaten raw, the use of untreated water to wash these foods can serve as a vehicle of transmission of pathogenic organisms. Ice is used to cool and maintain coolness in a variety of fresh foods. As the ice melts, the organisms associated with it are passed on to the foods and vice versa. The reuse of ice is neither a recommended nor an accepted procedure since this ice is contaminated. The use of water in a food processing plant can be a source of microorganisms for contaminating food. Water is the food industry's number one raw ingredient. Water enters into the processing of most foods for cleaning equipment and processing areas, for washing food, for conveying or flaming foods, or as an ingredient. The public health aspect. From the public health point of view, the water used for the food must be absolutely safe to drink as shown by freedom from sewage contamination. To test this, the water is free from sewage contamination is by counting coliform bacteria. The production of acid and gas in the fermentation tube of lactose broth at 35 to 37 degrees Celsius is the positive test for presence of coliform bacteria. The test may be confirmed by some other techniques. For example, for Escherichia coli, which is of intestinal origin, can be distinguished by plating on eosine methylene blue agar. If the metallic sheen is produced, it means that confirms the presence of Escherichia coli, and the Enterobacter aerogenes, it is from the plant surfaces at the soil origin. Frequent lab tests should be conducted. In case of any doubt, chlorination of water is done at the rate of 0.025 to 2 ppm or even more on the basis of available chlorine depending upon the composition of water and extent of contamination. The economic aspects. The water with agreeable chemical and bacteriological characteristics is desired. For processing of food, plenty of water of desired composition should be available. Water should have acceptable taste, odor, color, clarity, chemical composition and bacteriological composition. The desired chemical composition is affected by hardness, alkalinity, organic matters present, iron, mineral and fluorine. The different methods of water purification. The water source should be protected from sewage contamination. It may be purified by sedimentation in reservoirs or lakes, filtration through fine filters or sand or by chlorination, ultraviolet irradiation, or by boiling. Only partial purification is achieved by sedimentation. Efficient filtration greatly reduces the microbial content, but filters can be a source of contamination of water with undesirable bacteria. Filters for soft drinks sometimes add large number of coliform bacteria. UV irradiation has been found satisfactory. Now, the another important source of contamination is air. Today, the main concern with air pollution is with chemicals, that is carbon monoxide, hydrocarbons, soot, fly ash, rather than with the biological agents. Nature is the major contributor of not only the chemical pollutants, but of biological agents such as plant cells, animal hairs, pollen, algae, protozoa, bacteria, yeasts, mold spores, and viruses. Food is subjected to airborne contamination until it is sealed in a package. The adjacent figure shows the different mode of air contamination to the food. The types and numbers of microorganisms. There is no natural or normal microflora in air, which is contaminated from various sources. Generally, mold spores are more prevalent than are other microorganisms. The main source of microorganisms, especially mold spores, decaying plant materials near the ground surface. Contamination of the air is caused by gusts of wind picking up the microorganisms or spores. Compared to mold spores, there are relatively few yeast cells in the atmosphere, and these are mainly near ground level. One would expect yeast to be prominent in the air of orchards or vineyards. However, even here they may comprise only 25% of the fungal population of the air. There are many sources of microorganisms for contamination of the air. Liquids are aerosolized or by spraying, splashing, the bursting of bubbles by forcing through a small orifice or by vibrations. Trickling filters used in sewage treatment. Trickling filters used in sewage treatment plants produce aerosols by the spraying action and by the splashing of liquid sewage. The application of wastewater to land by spraying under pressure causes aerosolization of organisms. The types of organisms in air are often associated with the types of activity in the area. For example, streptococci are found near dairies and yeasts are found near bakeries and breweries. Human beings shed organisms and also produce aerosols during talking, coughing, and sneezing. The microbial load of the air is proportional to the number of persons in an enclosed space, their activity, and the rate of air circulation. Although humans might not contribute significantly the microorganisms, outdoor air they can be significant source in a processing room. Thus, air is subjected to a number of sources of microorganisms, for example, from decaying matter, water, soil, human beings, animals and sewage. The numbers and types of organisms present in the air are atmosphere dependent upon many factors such as tendency to settle out and maintenance of viability. The number of microorganisms in air depends on amount of movement, sunshine, humidity, location and amount of suspended dusts. Number may vary from less than 1 per cubic foot at a top of mountain to thousands in very dusty air. Microorganisms settle down in quiet air and conversely moving air brings more microorganisms in. So, movement of people, ventilation and breezes increases the number. Direct sun rays kill microorganisms and reduce number in air. Dry air contains more organisms than moist in similar condition. Rain or snow removes organisms from the air. So, hard and steady rain practically free the air from microorganisms. According to a study of airborne contamination of food, it was stated that the microbial population of different processing plants are similar. Propagation vary tremendously in number from one part of a given plant to another. Population vary tremendously in number from one part of a given plant to another. Population in a plant are related to the air quality outside the plant and population levels are related to the levels of activity of workers. Now, the treatment of air. Microorganisms can be reduced by natural conditions, for example, sedimentation, sunshine, washing by rain or snow, or maybe artificial means, for example, filtration, chemical treatment, heat, electrostatic precipitation. The most commonly used method is filtration through fibers, for example, cotton, fiberglass, asbestos, activated carbon, etc. Fibers should be replaced periodically or sterilized by heat or gas. The washing by means of a water spray. By bubbling air through water is not efficient because air may get contaminated with the organisms present in the water. Chemical treatment is comparatively popular. Passage of air through tunnels fitted with UV lamps. Electrostatic precipitation of dust particles. Heating to a high temperature is efficient but expensive. During air is sterilized, precautions must be taken to prevent recontamination by positive pressure of air inside the room, a regular cleaning or washing of filters in air conditioning or ventilating systems, or UV irradiation air locks at doors reduces the number of organisms carried in by workers. Now, another important source of contamination is plants. Plants are contaminated by microorganisms from several sources like dirt, water, air, fertilizer, animals and humans. Once contaminated, certain microorganisms can grow on the plant surfaces and plant pathogens can attack their host plants. The microbial flora on plant surfaces varies with the kind of plant. Pseudomonas species are quite prevalent on vegetables. More flowers of fruits are inhabited by The flowers of fruits are inhabited by many genera of yeast, including the ascospores farmers, for example, Saccharomyces and Hansenula, as well as the imperfect, for example, Torulopsis, Candida, Rhodotorula and Cloicara. In some fruits such as grapes, the yeasts might not be present on the flowers, but are found on the ripe fruit. There is evidence that not only the surface, but also the interior tissue of plants can be contaminated. The number of bacteria will depend upon the plant and its environment and may range from few hundred or thousand to millions per centimeter square surface area. For example, inner surface of cabbage will have less and fewer kinds of natural microorganisms. Vegetables can harbor fecal streptococci within unopened pots, heads and other structures. Thus, live plants can serve as a source of microorganisms. When the plant dies, the decaying vegetation becomes an important source of airborne, waterborne and soil microorganisms, which then contaminate plants the following year. The another source of contamination is from animals. The animals have a normal or natural microflora that is established very early in life. Besides this microflora, animals tend to harbor the types of organisms found in their environment, since they are contaminated by soil, water, air, feed and excreta. Animals harbor organisms that can cause food spoilage or foodborne diseases. Organisms are found in animals' gastrointestinal tract, nasal passages, cutaneous lesions and on the skin, feet, hair or feathers on the outer surface. These organisms are readily transferred to the edible portions of the animal during processing. These animals also carry potential human pathogens that may be transferred to the foods they contaminate. The muscle tissue of most live, healthy animals is essentially sterile. In a diseased state or after slaughter or after harvesting, in the case of fish, bacterial invasion of the muscle tissue can occur. Most of the microorganisms in tissue are thought to result from surface or intestinal contamination or from processing operations. One of the sources of microorganisms in the interior portion of animals is the lymph nodes. These nodes tend to filter out bacteria from the lymphatic system. Microbial counts of 10 to the power 5 per gram have been obtained from the lymph nodes, with many types of organisms being present. The hides of animals may contain from 10 to the power 3 to over 10 to the power 9 microorganisms per square centimeter. Although they do not penetrate the intact skin, these microorganisms are a source of contamination of the edible portions of the carcass during processing. The feathers and feet of poultry also carry heavy contaminating organisms from above sources. The skin of meat animals may contain micrococci, staphylococci and beta hemolytic streptococci. Staphylococci from the skin or from the respiratory tract may go to carcass and finally to the raw end products. The prominent organisms in the intestinal flora of both animals and humans are obligate anaerobes such as species of bacteriocytes and Peptostreptococcus. The last is the sewage. Animal manure or in some cases human wastes are used as a fertilizer on crops. These biologically produced substances contain microorganisms including human pathogens. When added to soil, these pathogens may survive for periods sufficient to contaminate the harvested crop. In rural areas, septic tanks are used. Quite often these are not properly installed and do not operate effectively, and raw sewage leaks into the soil. Now, this is the flow chart how the sewage sludge contaminates our soil and then ultimately to the food. Here the plastics, pharmaceuticals and other harmful chemicals, they are transferred to the water or the sewage and wastewater contaminants. From there in form of micronutrients, although they are subjected to treatment, wastewater plant treatments, but still they contain some of the living microorganisms, and the leftover material, the sewage sludge, that is transported to the farmlands as a fertilizer, and there they will contaminate the soil ecosystems, and from there they will contaminate to the plants growing on that, or they will percolate to the water in the groundwater, and from there when the plants are grown on that, that will contaminate the food. Likewise the cycle completes. The salmonella are quite prevalent in raw sewage. Processing in sewage treatment plants lowers the incidence, but even at 99% reduction, if the original level of salmonella is high, they remain in the effluent and sewage sludge. Enteric viruses survive the treatment process, may remain ineffective when bound to solids, and can contaminate soil and groundwater when sewage sludge is applied to agricultural land. Natural water contaminated with sewage contributes their microorganisms to shellfish, fish and other sea foods, which in turn go to human being consuming this. Treated sewage going onto soil or into water contributes smaller number and fewer pathogens than raw sewage. That is all for today. Thank you very much.